We're going to start off by looking at the structure of a muscle, and we're going to go from big to small. So we're going to keep zooming in. The first thing to notice about a muscle is that it's tubes inside of tubes inside of tubes. Think of like those nesting dolls where you open one up and there's another inside, and then you open that one up and there's another one inside, and it just keeps going on and on. These tubes also have connective tissue that encloses them. So we're going to start off with the muscle as a whole. So here is our entire muscle. Now, these bottom sections of the picture are where they're zooming in on parts so we can see it more closely. But here we have the muscle as a whole. And it is enclosed in a connective tissue wrapping called the epimysium. So look for patterns in words. The epi means on top, and the my refers to muscle. Then that has tubes inside of it. So we have these bundles you can see. And I'm not going to outline all of them, but you get the idea here. Each of these bundles is a fascicle. And the fascicle is surrounded by connective tissue called perimysium. So you can see kind of all this connective tissue in between here. That is perimysium. And I'll explain fibers in a minute, but a fascicle is a bundle of fibers, and it is wrapped in perimysium. Notice also this is where you have arteries, veins, and nerves. They're embedded in the perimysium. Okay, then if you look in the fascicle, you can see more little bundles. So we have little circles inside here. And you can see it here also. These are the fibers. So if we come out to where they're zoomed in, they're zooming in on an individual fascicle. And then within that fascicle, we can see the fibers. And here is zoomed in on an individual fiber. A fiber is a muscle cell. So when we're dealing with muscles, we tend to say a muscle fiber instead of calling it a muscle cell, but it is just a cell. And muscle cells are long tubes of protein. And most cells in your body only have one nucleus. Muscle cells are the exception. They have multiple nuclei. The reason for this is when you were an embryo, you started off with a bunch of short muscle cells. So they were like this. And each one had its own nucleus. Well, now as an adult, your muscle cells are very long. The way you got these long cells was these individual ones fused into one long cell. And you kept all of those nuclei. So that's why muscle cells have multiple nuclei. They're actually several cells that fuse together. And the fibers have a connective tissue around them called endomysium. Now there's two coverings on a fiber. Endomysium is your connective tissue that wraps the fibers. Like any cell, a muscle fiber also has a membrane. And with muscles, there's a lot of things that we give it unique names. So just like we don't say muscle cell, we say muscle fiber. When you're talking about muscle fibers and their membrane, you don't say the cell membrane or the plasma membrane like we usually do. Instead, it's called the sarcolemma. Okay, so here's starting off the tubes inside of tubes. You have a muscle as a whole. It has epimysium, connective tissue surrounding it. That has long tubes called fascicles, which have perimysium wrapping them. Within a fascicle, you have these long tubes called fibers, which are wrapped in endomysium. Now, within the fibers, you can see there's more tubes. We can keep going with this whole tubes inside a tubes idea. So see, there's all these little dots here inside the fiber. These little dots in the fiber are the myofilaments. These are the tubes of protein that make up the majority of a muscle. And there's several types of protein in a muscle. The main ones that are these tubes of protein are actin and myosin. So now we're going to move on to the next picture, and we're zooming in. So this is an up close of a fiber. So you can see here the fiber level, right here, this circle, this circle. Now we're zooming in up close. So this right here, fiber, is what we're looking at up close here. So remember, we have the sarcolemma, that's the cell membrane. And we talked about the multiple nuclei, so that's all these purple things here. There's another one here and another one here. The little yellow things that you see are mitochondria. Right, there's one here and one here. Those, remember, are making ATP. We have a few other individual or unique structures to muscle. Notice these green things. These green things here are a T-tubule. Okay, so your nerves stimulate your muscles to contract, and nerves use electric signals. Your muscle also has to be able to conduct that electric signal. Electricity is what makes muscles contract. So one thing that is unique about your sarcolemma is that it conducts an electric signal. It will have an action potential just like an axon. So an axon is a part of a neuron that conducts an electric potential, and the sarcolemma is going to do that. It's going to have action potentials like neurons do. Notice also there are these holes. You have these pores, and those pores line up with the T-tubules. So the purpose of a T-tubule is it's a tunnel to let that electric signal down into the muscle. Then next to the T-tubule, there's all this purple stuff. This purple webby stuff is sarcoplasmic reticulum. This stores calcium. So if you think back to chapter 7 when we learned about bones, we talked a lot about maintaining blood calcium in that chapter. And one of the reasons you have to maintain blood calcium is your muscles need it in order to contract. So your muscles take calcium from your blood and they store it in this sarcoplasmic reticulum. Notice at the edges of the webs, you have these flat areas that are pressed right up against the T-tubule. That flat end of the sarcoplasmic reticulum is called a terminal cisterna. Now there's actually a little tiny microscopic gap. They're not pressed together, but they're very close to each other. So this is the flat end of the sarcoplasmic reticulum that is next to the T-tubule. And notice how they are all in these sets of three. It's kind of like a sandwich. So you get what is called a triad. A triad is the one T-tubule with a terminal cisterna on each side, just like a sandwich. You have the peanut butter in the middle and the bread on each side. And that terminal cisterna is filled with calcium. Okay, then if we get to the tubes, so we have here, let's look closely at these tubes. So this tube is a myofibril. So this is a myofibril, and then within the myofibril we have our actin and myosin. And the actin in our textbook is shown as red, and the myosin is yellow. Okay, these also go by thick and thin filament. Myosin is thick. So of these two proteins, myosin is the thicker one, so it is also called the thick filament. Actin is the thin one, so it is also called the thin filament. So back up here, it would be myofibril, then myofilaments. So your tubes go the entire muscle, the fascicle, the fiber, the myofibril, the myofilaments. Okay, so now you know the main parts. Now we can start thinking about how these parts work. The function of a muscle is to contract. So think back to what you've learned, the basics of muscles. And like in the study of tissues, you have a flashcard, muscles contract. Well, that means that they get shorter. If something contracts, it gets shorter. So think about body movement. When your muscle contracts, it gets shorter and it pulls on a bone, and that makes the bone move. That's how your body moves. Right? And you just learned that in chapter 9, like with the elbow model where you had the rubber bands. When the rubber band gets shorter, it pulls on the bones of the forearm and moves them. If we look here at our myofibril, you have right here this zigzag line, and you have another zigzag line right here. Those zigzag lines are Z-disc. And you have another one here. The area from one Z-disc to the next is a sarcomere. So everything in between these two Z-discs is a sarcomere. And that repeats, so it goes on all along the tube. So then you would have another sarcomere here and another sarcomere. Like you can see, here's a Z-disc, here's a Z-disc, that area is a sarcomere. Then there would be another one here, and so on, all along the length of that tube. The way a muscle gets shorter is each sarcomere gets shorter. And if all those sarcomeres get shorter, the whole fiber gets shorter. And when the whole fiber gets shorter, that makes the muscle shorter. Okay, so we just talked about sarcomeres. Here is some sarcomeres up close. Remember, it goes Z disc to Z disc. So here, these zigzag lines are your Z disc. So here's a Z disc, here's a Z disc. So this area from one Z disc to the next is a sarcomere. Okay, and then here's another Z disc. So here's sarcomere number 2. Here's another Z disc. Here's sarcomere number 3. So this picture shows 3 sarcomeres. And in real life, you would have hundreds of them lined up end to end. Okay, so now that we're zoomed in on a sarcomere, and just to go back and show you how it relates to the previous picture, remember, here's a sarcomere, the area in pink. We're going to look at the parts of the sarcomere. So you already know there's actin and myosin. This is the actin. Actin is in red. And this is the myosin. The myosin is in yellow. Okay, there's some other parts to label here. There's this brown line in the middle. This is the M line. The M line is like the anchor. It's not going to move. So this is our anchor point. We have this green stuff is connectin. Notice it is coiled up like a spring. So it's going to compress and then release. This is what pushes the sarcomere back out to full length when you're done contracting the muscle. It's a spring that gets compressed when the muscle contracts. Okay, if you look closely on the actin, there's a green line that wraps around it and then those blue dots. Now remember, all of these colors are just colors that the artist of our textbook picked in order to make this easier to see. It's not these colors in real life. But we have here this green line that wraps around the actin is called tropomyosin. It is a regulatory protein. Its job is to prevent the muscle from contracting. You don't always want your muscles to contract. They have to alternate between contracting and relaxing. If they just contracted and stayed contracted, you would have rigid paralysis. So you have to also relax. So this protein is involved in making the muscle relax. Then there is this blue dot on there. That blue dot is troponin. Some of you may have heard of troponin before. If somebody's had a heart attack, they find lots of troponin in the blood. The heart is a muscle. The heart also has troponin. And when a muscle gets injured or damaged, the troponin breaks off and it floats out into the bloodstream. So that's true of skeletal muscle also. If you get an injury to your skeletal muscle, you're going to have high levels of troponin in your blood. So that's how they can test somebody to see if they've had a heart attack, is they do a blood test and see if there's a lot of troponin in the blood. Okay, so those are the basic parts of a sarcomere. Now we know that the sarcomere is going to get shorter. And we know that the borders of the sarcomere are the Z disc. We know that the M line is the anchor. So the basic way that a sarcomere gets shorter is you have to move the Z disc in toward the M line. So we want the Z disc to go this way toward the M line. When they do, the sarcomere is shorter. So we're going to make this shorter. Okay, so now let's think about how we're going to do that. How do we get the Z disc to go to the M line? Well, notice that the actin is connected to the Z disc. So if we have the actin move toward the M line, the actins will pull the Z disc toward the M line. Okay, so let's keep going. Let's ask now, well, how do we get those actins to move toward the M line? Well, let's look at the myosin. Notice the myosin has all these little bumps on it. Those are called the myosin heads. So each little bump here is a myosin head. Think of those like hands. Think of you have a rope and you're moving that rope. You're on a boat and you have an anchor attached to a rope and you want to pull the anchor in. You use your hands to grab the rope and pull it in. That's what the myosin heads do. They attach to the actin and they pull it toward the M line. So the way we get the actins to move in is the myosin heads attach to the actin and pull it toward the M line. Okay, so now let's review what we know so far. We know that the muscle contracts means it gets shorter. We need the sarcomere to get shorter. So the myosin heads bind to the actin and they pull the actin in toward the M line. And the actin is attached to the Z disc, so when the actin moves toward the M line, the Z disc moves toward the M line. And when you do that on both sides, the sarcomere gets shorter. You end up with the Z disc there at the tips of the myosin. And that connectin, remember, is a spring, so it can compress. So that connectin just gets compressed as this happens. So let's look at a picture. Here's a picture of a contracted sarcomere. So you can see here it is at the top when it's relaxed. You have that gap there between the tips of the actins and the Z disc are out far from the M line. You're going to have the myosin heads bind to the actin, pull the actin toward the M line. The actin is attached to the Z disc, so it pulls the Z disc to the M line. And this picture is what you get. See, the connectins are compressed. You've got the actins there overlapping at the M line, and you've got your Z disc just right there, just barely away from the tip of the myosin. And that sarcomere is shorter. Okay, so now that hopefully you understand that, let's add a few more details. Remember, we have to regulate this. So if it were up to the muscle, it would contract all the time. The myosin always wants to pull the actin in and contract. We have to have a way to control this. You only want it to happen when your brain decides that your muscle should contract. So the basic setup here, the muscle always wants to contract. When your brain wants the muscle to be relaxed, you have to stop it from contracting. Then when your brain wants the muscle to contract, all you do is let it go. You stop inhibiting it. I think of it like a dog that's really, really energetic and rambunctious, and you put that dog on a leash to go for a walk, and that dog just wants to run. And the whole time, like, you're holding tight to the leash, holding back on that dog. Then when you want the dog to run, all you have to do is drop the leash, and the dog will take off. That's how your muscles are. They want to contract. You rely on that tropomyosin to make them relax. Then when your brain is ready for the muscle to contract, all you gotta do is get the tropomyosin out of the way and the muscle will contract. Okay, so let's look in some more detail now at how this is happening. When the muscle is relaxed, you have tropomyosin getting in between the actin and myosin. If we look at the picture here, see the tropomyosin is that green strand. It wraps around the actin. And see the little black dots on actin? Those little black dots are the active sites. That is where the myosin has to bind. You can see over here in picture 3B, the myosin binds right on that black dot on the actin. Well, when the muscle is relaxed, the tropomyosin is covering those active sites. So let's go ahead and label that. That is an active site. So when the muscle is relaxed, tropomyosin covers the active site on actin. As long as this site is covered, that means the myosin head cannot bind. And therefore, myosin can't pull actin in and the muscle can't contract. So the muscle is relaxed. Now let's look at what happens when your brain wants the muscle to contract. Your brain's going to send a signal through a nerve. And that nerve is going to cause an electric signal on the sarcolemma. So remember, sarcolemma is the membrane of the muscle cell. That electric signal will go down the T-tubule. Okay, remember what's right next to the T-tubule? Sarcoplasmic reticulum, and terminal cisternae are the flattened ends of the sarcoplasmic reticulum. When the electric signal is in the T-tubule, it causes that terminal cisternae to open its calcium gates. So the electric signal in the T-tubule stimulates the terminal cisterna. And if you need to, go back and look at the picture of that. We'll just go ahead, we'll look. Right there, the T-tubule. Remember, it has a terminal cisterna on each side. When there's an electric signal in that T-tubule, it makes that terminal cisterna open its calcium gates. Okay, think back to now chapter 4, what you learned about diffusion. If it's all full of calcium in there, that calcium wants to diffuse out. Remember, it wants to spread out until it's equal. So as soon as you open the gates, the calcium diffuses out. Okay, in this picture, that little tan dot is the calcium. And do you remember what the blue thing is? The blue is troponin. Notice the calcium binds to the troponin. So once the calcium diffuses out, it binds to troponin. Troponin is attached to the tropomyosin. So once you have calcium binding to troponin, then the troponin moves the tropomyosin off of the active sites. So that's why in this picture, the active sites are exposed. The tropomyosin has been moved by the troponin because calcium bonded to it. So once calcium binds to troponin, then the troponin moves the tropomyosin. Now that tropomyosin is out of the way, actin's active sites are exposed. So remember, the muscle is like an energetic dog that wants to take off and run. And the tropomyosin is the leash that is holding that dog back. Once you drop the leash, the dog takes off. So your muscle always wants to contract. You inhibit it with tropomyosin because it blocks the myosin from being able to bind to the actin. Then when your brain sends a nerve signal, the nerve signal goes down the T tubules. It makes the terminal cisternae open their calcium gates. The calcium diffuses out. Calcium binds to troponin. The troponin moves the tropomyosin. So basically, the calcium is a signal saying, hey, drop the leash. And once you drop the leash, now the myosin is free to bind to the actin, pull it in, and the muscle's going to contract. So let's look at how that happens. So here we have what is called cross-bridge formation. Once the tropomyosin is out of the way, the myosin head binds to the actin. So we don't go around saying myosin head binds to actin. We say a cross-bridge has formed. So if we go back over here and we keep writing this on our list, once the actin's active sites are exposed, we form a cross bridge, which means the myosin head binds to the actin. Okay, now notice the angle. The myosin head binds to the actin like this, at this angle. Then it bends to this angle. Well, that's how you get the motion. That's how you move the actin in toward the M line, is when that myosin head bends. And when the myosin head bends, that is called power stroke. So it binds out at an obtuse angle, then it bends to an acute angle, and that's what pulls the actin in. So after you form a cross bridge, then you power stroke. Okay, this also is when you use your ATP. Remember when you use ATP, it breaks into ADP and PI. So right here, this is ADP and PI, and then here it is released. And remember, this is going to go back to the mitochondria to be reattached. Think back to chapter 3 for that. But every time you do a power stroke, you use an ATP. Okay. Now think about back to being on a boat and pulling in an anchor. You can't just grab the rope and pull it once and all of a sudden the anchor's in. You have to repeat, right? So one hand grabs the rope and pulls. Then while the first hand is holding the rope steady, your second hand reaches out and grabs the rope further down. And then it pulls and your hands keep taking turns. One little power stroke is not going to pull that actin all the way in. Notice you have several myosin heads there. That's like your two hands pulling the rope. They have to, one binds, cross bridge, power strokes, which pulls it in just a tiny bit, and then another one has to form a cross bridge and repeat this process. So just like with pulling in the anchor in your boat, Once the second hand grabs the rope, the first hand has to let go so it can extend back out. It's the same thing here. You have to keep repeating. So the next thing that happens is we have to break the cross bridge. Now, this one is breaking the cross bridge. While it's doing this, another one is back here in step A, forming a cross bridge. You have multiple myosin heads. So while one's breaking a cross bridge, another is forming a cross bridge. Okay, and in order to break the cross bridge, you have to have a new ATP binds. If you don't have a new ATP come in and bind, you get stuck with that cross bridge. And that's actually why rigor mortis occurs. When someone dies, the body gets stiff because without that control from the brain, you release all the calcium and the muscles all form cross bridges and power stroke, but then you stop making ATP once you're dead, so no ATP comes along to allow the breaking of the cross bridge. So rigor mortis is that stiffness of a dead body because all the muscles get stuck right here in step 3. They power stroke, but then they can't break the cross bridge without new ATP coming in. So anyway, an ATP binds, you break the cross bridge, which just means the myosin detaches, the myosin lets go. And then the myosin resets. So notice the angle. We're at acute. It resets back out to the obtuse angle. And then look at our arrow here. It leads us back. We repeat. We keep repeating that until the actins are all the way in. So you power stroke, then you break the cross bridge, then you reset the myosin, and then you come back here and you repeat with forming a cross bridge. And by the time you've repeated this a few times, the Z disc are moved in here close to the tip of the myosins, and your sarcomere is shorter. So remember, here's how we started. This one is relaxed. It's a long sarcomere. The myosins bind to the actins, cross bridge formation, they power stroke, they pull the actins in toward the M line, and now you have a shorter sarcomere. That's how muscles contract. Okay, so one last picture here is to just kind of put all that together, go over it in the big picture, and talk about how you get back to relaxed. So this is just a summary. So here we have, this is our neuron. And here it is secreting neurotransmitter, which is acetylcholine. And you can abbreviate this ACH. This is the neurotransmitter that makes muscles contract. And when you get that acetylcholine secreted on the muscle, you're going to have an electric signal, which is called an action potential. Remember that electric signal moves along the sarcolemma. Which is the membrane of a muscle cell. Here you can see it goes down the T-tubule. And then here you can see all the calcium diffusing out of the sarcoplasmic reticulum. So it's having an electric signal in the T-tubule is what stimulates the terminal cisterna to open its calcium gates and let the calcium out. The calcium diffuses down here where it binds to troponin. That makes the tropomyosin move out of the way. Now you have the actin active sites exposed, so you can form a cross bridge. Then the myosin head will bend, which is power stroke. This pulls the actin in toward the M line. So you can see our arrows here. You pull in toward the M line. Then you have to break the cross bridge, reset the myosin, and then repeat until you get the actins all the way pulled in. And then remember, the actins pull the Z disc in. And now your muscle is contracted because that sarcomere is shorter. Okay, but now you also have to relax, right? You don't just want that muscle to stay contracted the rest of your life. You have to be able to relax it. So now let's look at how you relax. In order to relax, you stop sending the signal. So remember, your brain's in charge. So your brain will stop the signal. And once the signal stops, there's no more acetylcholine. So then there's no more action potential. And if there's no action potential on the sarcolemma, it's also not in the T-tubule. Now you close the gates on the sarcoplasmic reticulum and the terminal cisternae. Okay, then remember back in chapter 4, pumps that move substances against their concentration gradient and make them more concentrated. So now you will use calcium pumps in the sarcoplasmic reticulum to gather all that calcium back up. Think of it like a million little shop vacs. They go out and they vacuum up all that calcium back into the sarcoplasmic reticulum. So you take the calcium away from the troponin. And once you take the calcium away from the troponin moves the tropomyosin back to blocking the active sites. And once that tropomyosin is back over the active sites, you can't make any more cross bridges. So this means the myosin and actin cannot attach to each other anymore. And once they can't attach, that connectin springs back out. So remember, our connectin is these little green coils at the end, and they're like a spring. Once the myosin and actin can't hold on to each other, then that spring pops back out, and it pushes the Z disc back out to their resting position. So we say it pops or it kind of springs. So it pushes those Z discs back out away from the M line. So now the sarcomere is long again. And the muscle is relaxed. Our next topic is motor units. Take a look at these motor neurons. These are the neurons that are going to stimulate the muscle fibers to contract. Notice we have 3 neurons here, and they're color-coded. When they get down to the muscle, they branch, and those branches go out to different muscle fibers. So you have multiple neurons to a single muscle. And each neuron branches to multiple fibers. A motor unit is one neuron and the fibers it branches to. And whenever that neuron gives a signal, all of the fibers in the motor unit contract. So you can think of it as teams. Think of your muscle as being divided up into teams of fibers. And all the members of a team contract together. So if the purple neuron has a signal in it, all of the purple fibers down here will contract. If the red neuron has a signal, then all of the red fibers contract. There's several important features of the motor unit. One is the distribution of team members. If you look back up here at the picture, notice they're not together, they're spread out. Look at the blue one, for example. The blue fibers are all spread out from each other. They're not lumped together. Think of that as 2 people trying to move a couch. If 2 people are trying to move a couch and they both get on the same side of the couch, they're not going to move it very well. That's not a good distribution of force. If they spread out from each other and get on opposite sides of the couch, they distribute the force and they're going to work. They're going to be able to move the couch. So the distribution of the fibers in a motor unit is that the fibers of a unit are spread out from each other. That way, when those fibers contract, you get an even distribution of the force. Another important feature of motor units is the size of the motor unit. All motor units have one neuron, but the number of fibers can vary. So a small motor unit is like a basketball team. Think of the neuron like the coach and the fibers like the players. So a small motor unit has only a few fibers per neuron. A large motor unit is like a football team. You have many fibers per neuron. And in a human body and the muscles, could be several hundred fibers per neuron. So there's advantages to each. Think about a small motor unit. Think about a basketball team. If one neuron has only a few fibers to control, you can get very precise movements. This is what gives you your fine motor skills. So in the muscles that control your hands and your fingers and your eyes, you have very small motor units because you have very precise fine motor control. Okay, now think about a large motor unit. One neuron gives a signal and hundreds of fibers contract. This gives you a lot of power. You get a huge response just from one signal. This is found in your legs because your legs have to carry your body around and they need a lot of power in order to do that. Another feature that we get from motor units is the degree of force, or we get control of the force. You don't necessarily have to use all your motor units. Your brain can decide how many it wants to use. Suppose that you are babysitting a baby and you're at the park, it's a nice day, but a mosquito lands on the baby's nose. You would use your arm to gently shoo the mosquito away. What if you're at the park and a hungry bear comes charging at you? This time, instead of gently shooing the bear away, you're going to use your arm to punch the bear as hard as you can. It's the same movement, but one time you can do it gently, one time you can do it with all your might. So we have here the all hands on deck, right? This is when you want all your might. You want lots of force. This would be punching that hungry bear. In this case, you would use all of your motor units. So say the muscle, like the one in our picture up top, was split into 3 motor units, you would send a signal through each of those neurons and have all 3 motor units contract. Okay, over here we have a skeleton crew, right? Which means you're just doing the minimal force. This is when you're shooing the mosquito from the baby. You want to be very gentle and careful. So here you're only going to use 1 motor unit. And you can do anything in between. Suppose you have a muscle with 10 motor units and you need a medium amount of force, maybe you'll use 5 of them. So you can always control how much force you use by how many motor units you stimulate. And the final thing about motor units is that they take turns. So individual fibers get tired. And that is called fatigue, when they get tired. And they need to rest. But the muscle as a whole can stay contracted for a long time. This is because the motor units within the muscle take turns. Suppose you're sitting in class with your lab partners, and I say that you'll get a bonus point if you can hold your book up in the air the whole class period. Well, your arm is going to get tired after a few minutes, but you don't want to set the book down because then you lose. So you ask your lab partner to take a turn holding it, and when that person's arms gets tired, they ask the other lab partner to take a turn. If you go through and you each take a turn holding the book up, you'd be able to keep the book in the air for the whole class period. That's how your muscles are. This is especially true in your back. That's where your posture muscles are. They have to work all day long. And the way they keep you upright all day long is the motor units within those muscles take turns. So there is everything you need to know about motor units. Now we're going to see how muscles get energy and types of muscle fibers. Let's think about what you learned in Chapter 3 about ATP. Remember that ATP stands for adenosine triphosphate. So it's an adenosine with 3 phosphates attached. And that third one is attached by a high energy bond. Hopefully this is coming back to you from our Chapter 3 lessons. When you need energy, you break that bond. After you break it and the cell gets the energy, you are left with ADP, with the D standing for di for 2. And then that phosphate you broke off is by itself. Then the cell regenerates this. So remember, these 2 things go to the mitochondria and you reattach that phosphate. And you're back to having ATP. So the basics of getting ATP is you just attach a phosphate to an ADP. Muscles have multiple ways to do this. In Chapter 3, you learned about aerobic cellular respiration, where you do glycolysis and the pyruvates enter the mitochondria, you do Krebs and electron transport, and you attach the P to the ADP. You also learned about fermentation, when you go into oxygen debt and the mitochondria shuts down and you just do glycolysis. So you already know 2 methods. In muscles, there's 4 methods. So we're going to take those 2 you already know, and we're going to add some more. This picture of the track is showing you when you use each of those 4 methods. We're going to start with the pink one. So notice the pink is the first part of the track here. Here's your start line. The pink is the one you use when your muscles first start working. And this is the phosphagen system. So phosphagen is the immediate energy when a muscle first starts working. Notice, though, how short-lived it is. It's only those first few strides of running around the track. So it's immediate, but it is short-lived. Next, we go to this orange one. So you can see here, here's runner number 2. he's continuing on around the track like this. The orange one is anaerobic. Aerobic means oxygen, and when you put a or an in front of the word, it means without. So anaerobic, you're not using oxygen. Okay, this is what you switch over to after phosphagen. So you can see even runner number 2, he started in phosphagen. Then as he kept running, he switched to anaerobic. Number 3 is yellow. So I'm going to use kind of this tan color instead. Here's runner number 3. And see, he started here in phosphagen. Then he went to anaerobic. Then as he comes around about to this part of the track, he switches over to the yellow. This is the one you already know. This is aerobic cellular respiration. This is the one where you use oxygen and you get lots of ATP. This one produces the most ATP. Then the fourth one, it's not shown on the track here, but eventually you would go into fermentation. This is when you would have oxygen debt and you're forced to go into fermentation. You can't do aerobic anymore because your oxygen is not there to supply it. So that's an overview of the 4 steps. So you go through these 4 in order. So now we're going to look in more detail at each. So here is the phosphagen system. This is also sometimes referred to as phosphate transfer. Okay, the first part of this, it has 3 components. The first part, ATP already present in muscles. So when you first start using a muscle, when it goes from resting to being used, you're going to use ATP that's already there. The second part is where you use an enzyme called myosin kinase. So this is an enzyme. Okay, enzymes are going to build or break molecules. They're building and breaking bonds. Here's what you start with. You have 2 ADPs. So that's an adenosine with 2 phosphates. And we have another one. The myosin kinase enzyme comes along and you end up with an ATP, which is what we want, and then AMP. In this case, the M stands for mono, which means 1. So hopefully this is pretty simple to figure out. All this enzyme did was take a phosphate from one ADP and give it to the other. So you started out with 2 adenosines, each with 2 phosphates. You take one away, give it to the other. Now you have an adenosine with 3 and you have an adenosine with 1. Our third one is ADP. So we're going to have an adenosine with 2 phosphates, then creatine phosphate. Then this enzyme, creatine kinase, comes along, and then you have an ATP and just creatine by itself. Same idea. The enzyme took a phosphate from here and gave it to the ADP. This last part here is showing resting muscle. So this is what you do when you're not using the muscle. It regenerates its creatine phosphate. So when a muscle's resting, it will use ATP. It will take a P off of the ATP and give it to the creatine. So then you get ADP and creatine phosphate. Then when your muscle's working again, you're ready to go with this. You may have heard of creatine as a supplement. If you look at like the muscle milk that bodybuilders use, it is a supplement to help with building muscle. Okay, there's nothing special about creatine phosphate that makes muscles grow, but what it does do is give more energy for lifting weights. So the idea is it enhances your workout because you have more energy and you can lift more weights. I think of that South Park episode where Cartman took the weight gain 9,000 because he thought he would get big muscles, but all he did was sit on the couch and eat cheesy poofs after his weight gained 9,000 and he just got fat. He didn't get big muscles. So there's nothing magical about it that'll make your muscles big. You have to do the workout lifting the weights, and it just gives you more energy to do that. All right, then the next one is anaerobic. This is where you just do glycolysis. So you can see you take glucose either from the blood or you get glucose from glycogen stored in the muscle. If you remember back to chapter 2, that chapter 2 post-class assignment, when you have extra glucose, your muscles will store it as glycogen. So you can also get glucose from glycogen in the muscle. Glycogen is just stored glucose. Then you're going to break the glucose into 2 pyruvates. And you get your 2 ATP. So this is a pretty fast way of getting ATP. This is good for quick bursts of energy. Think of this like a firecracker. Anaerobic is great for things like sprinting and weightlifting. Things where you just need quick bursts of energy. Then we move on to aerobic. This is what you learned all about in chapter 3. This is where you do glycolysis. And you get your pyruvates. Then the pyruvates enter the mitochondria, where you do Krebs and electron transport. Okay, so here you can see you can get glucose from the blood, you can get glucose from glycogen. But you have other options. You also can get fatty acids and amino acids and send those into your mitochondria. So you have more options for your fuel source. And aerobic, the name literally means oxygen. So you also need oxygen going to the mitochondria. So as you can see here, you can take oxygen from the blood, or you can use your stores. So just like glycogen is stored glucose, you have myoglobin that stores oxygen in your muscles. So you can also get oxygen from the myoglobin in your muscles. So if we define myoglobin, it is a molecule in muscle that stores oxygen. So aerobic is going to give you lots of ATP. This is your big ATP producer. And how long you can keep doing aerobic varies from person to person. And if you remember back to chapter 3, it varies based on how much oxygen you have, which goes back to the health and efficiency of your heart and lungs. So you can keep doing aerobic until you go into oxygen debt. And this, how long this is, depends on your heart and lungs. So a well-trained athlete has really good heart and lungs that can do this for hours. Somebody who's out of shape is only going to be able to supply oxygen for maybe a few minutes, and then they're going to be done. Then our last one is fermentation. So everybody eventually will go into oxygen debt if you keep exercising. So fermentation is once your heart and lungs can't keep up with your muscles. So there's no more oxygen going to the mitochondria. So if there's no more oxygen, you shut the mitochondria down. Remember the moose out front? Says, sorry folks, park's closed. So there's the moose. The moose tells the pyruvates they can't come into the mitochondria. So now, because the mitochondria is shut down, you can only do glycolysis. And you're also starting to get lactic acid. You convert your pyruvates to lactic acid. And you only get 2 ATP, which is not very many. So once you go into fermentation, your muscles start to shut down. You're going to stop exercising. So those are the 4 ways that muscles can get energy. Then we can look at types of muscle fibers. So there's 3 types here. I'm just going to focus on the 2 ends, the 2 extremes, because the ones in the middle are just kind of medium on everything. So we're going to look at slow oxidative and fast glycolytic. So first we'll talk about slow oxidative. If you break the name down, slow, they're slow to get going on their ATP production. These are not the quick burst of energy. These take a little bit of a lag time. And oxidative means they use lots of oxygen. Another name for these fibers is slow twitch. If you're into sports, you've probably heard discussions of slow twitch and fast twitch fibers. These fibers specialize in aerobic. So most of their ATP comes from aerobic cellular respiration. So think about the features, the physical features they would need in order to specialize in aerobic respiration. Aerobic uses oxygen. So that means they're going to have lots of myoglobin because they use lots of oxygen. And myoglobin is red. So these fibers have a red color. And that's what the pictures up here are showing you. The dark red ones here, that would be a slow oxidative fiber. And since they use a lot of oxygen, they also have lots of mitochondria. So these fibers are using lots of oxygen, lots of mitochondria, and making lots of ATP. So they are fatigue resistant. This means they don't get tired easily. They can keep working for a long time. So this chart shows some other features. I'm not going to talk about everything in the chart. I went over a few highlights, and you can look at this chart for the rest of the features of slow oxidative. Now let's look at fast glycolytic. They are fast because they are fast to respond. And glycolytic means that they use glycolysis. So these fibers specialize in method number 2, the anaerobic. So they're using lots of glucose. So this means that they need a lot of glycogen. And since they're only doing glycolysis, that doesn't use oxygen. So they have a very low oxygen use. So since they don't need very much oxygen, they lack myoglobin. So this gives them a white color because of the lack of myoglobin. And you can see up here, these lighter colored ones would be the fast glycolytic. Also, since they're not using oxygen, they don't have much use for mitochondria because mitochondria is the part that uses oxygen. So they are low in mitochondria. These fibers have quick burst of energy. So they are quick and powerful, but they fatigue quickly. When it comes to the fibers, there's 2 things to take into account for how many you have. So the first thing is the function of the muscle. If we look back at the chart, we can see that slow oxidative tend to be found in trunk and lower limb muscles. Remember, these are the fibers that are fatigue resistant. They can work for a long time. So your trunk and your legs are what have to work for long periods of time. Your trunk is where your posture is, and you have to stay upright all day long. And your legs carry you around all the time. You stand, you walk. So they tend to have lots of slow oxidative. Here you can see for fast glycolytic, upper limb muscles. So your arms are mostly fast glycolytic. Your arms tend to work in quick bursts. You pick things up, but you don't walk around on your arms. You don't sit there all day holding things up above your head. So your arms work in quick bursts. And a lot of times when you're picking things up, it's heavy and you need a quick, powerful burst. So arms have a lot of fast glycolytic. So the function of the muscle is one factor in determining which type of fiber you have more of in that muscle. Genetics is another factor. So everyone has both types of fibers. But some people are born with more of one than the other. This is where natural talent comes from. If we go up to these pictures at the top, you can see here is our distance runner. This person has a natural talent for running marathons, and this person has lots of slow oxidative. So that's a genetic trait. He was born with a lot of slow oxidative fibers, so he's better at endurance activities. Here's our sprinter. If you look at the sprinter's muscle, you see lots of fast glycolytic. This gives him a natural tendency towards sprinting. So training, of course, is part of it, but the number of each type of fiber you have can give you a natural tendency toward one type of activity or the other. So there are your fiber types.